speaker is Dr. Allison Sgro. Us on an adventure.
I didn't realize I was promising an adventure, my goodness. All right, so I'm really interested in how groups of cells coordinate decision making to work together, and in particular, how they communicate to do this and how they encode information about their communication to decide how to behave as a group. And part of the reason I got interested in this was sort of watching people. People have a lot of different decision-making paradigms. The first one is a sort of a follow the leader paradigm, right? You can have some king tell you what to do, and that works sort of okay until there's a revolution, right? You can have a group discussion, and that may work okay in faculty meeting, maybe less well in Congress, depends on your faculty meeting, right? And people also engage in collective behaviors, all right? So if you're in a mosh pit or in a stadium wave, you can sort of behave almost like a physical particle. And part of what even got me to thinking about information was how we're using and processing information to engage in these kinds of behaviors. Cells have the same kinds of problem, right? So cells can be told what to do in your heart by a pacemaker cell that sets the pace of what they need to do, all right? You have to have a certain number of cells telling these cells what to do, or they can't actually coordinate your heart pumping. Cells also appear to engage in sort of these physical collective behaviors like our stadium waves, right? They're going to move in cell sheets on a surface. You can describe them like particles. It seems to be just a sort of physical forces kind of behavior. All right, but what I'm really interested in, I want to tell you a little bit about today and sort of give you perspective on how we can interrogate these and what the role of information coding and theory can be, is understanding cell signaling mediated discussions. All right, so what do cells do when they need to sort of talk among one another, partition things out, execute different programs, and they have many different choices on how to behave? All right, this is somewhere I think we can make a lot of progress, and we have a lot of new tools, and I want to give you sort of my perspective on what experimentalists can bring to the table, all right, to allow us to then understand how are cells encoding information and using this information to make decisions. All right, and so I want to sort of, I know there's a breadth of backgrounds in the audience, so I want to sort of give you an idea of what do I mean by things that cells can be using to decide to behave as part of a group. All right, so I want to sort of give you an idea of features, what the sort of inputs to these cells are, and how we can sort of visualize the outputs that a cell has. All right, so if you think about it, a feature of a cell could be its location in a group. All right, and we can actually control this in a lab now using a surface patterning or scaffolds. Right, so we can tell every cell where to go. All right, and cells can actually sense these their locations, and we are able to sense those locations ourselves using time-lapse microscopy. All right, and take lots of videos. So I'm in an after-lunch slot. You're going to get lots of videos. All right, cells also encode information using intra and intercellular signaling. All right, so we can see this intracellular signaling now. We can first encode it via microfluidics. So we've heard a little bit about that so far. Right, but we can sort of deposit and put little signaling molecules between cells to control their communication or turn on and off their own communication and interrupt it. And people are working on techniques to allow you to visualize this kind of output. I'm not going to talk about this today, but it's an area of active inquiry. So how do we actually see cell-cell communication and the information going between cells? All right, and finally, something I'm going to show you a lot of today is how do cells encode information in intracellular signaling. All right, we now actually have the ability to control this directly in a lot of systems. You may think of neural systems, but you can do this in other kinds of cells using a technique called optogenetics, where you can use a genetically encoded tool to go ahead, send light into a cell, and control what it does. All right, and we're able to visualize, and this is really what I'll focus on today, these behaviors using genetically encoded biosensors. So Eric talked a little bit about this earlier, this idea that cells might encode information in dynamics, right, and that it's possible now to read these out. I sort of want to show you what that looks like and what the sort of state of the art is in extracting information from these systems. All right, so my lab works at a couple of different systems. We work on cellular slime molds, tissues, and bacterial swarms. And part of the reason we do this is because I'm really interested in the idea that we can basically go ahead and work all the way from a system that's using molecular level signaling on a time scale of seconds, using that to coordinate processes across a cell on a time scale of minutes, to coordinate group-wide behaviors over hours and perhaps even days. All right, this is the big challenge facing us. How do we go ahead and understand how these seconds and minutes time scale dynamics coordinate processes that happen over hours and days? So I'm going to start here. We might get to tissues, but at the very least, come talk to me. We work on all of these different things. It's a really broad problem. I think it's why we're such a broad audience as well. All right, so cellular slime mold does this really cool thing. When they're starving, they execute this behavior where instead of working by themselves, they join together into groups. So what's going on here is there's a horde of slime mold eating bacteria over here, and as this horde runs out of food, they turn on this program where they send each other this extracellular signal. Receiving it causes them to round up, and that's why you see these waves, and they aggregate together in these groups. And in these groups, they're going to differentiate into two different cell types. About 20% of the cells are become the stock cell. This is sort of a almost 3D video. Yep, they're putting this up now. But 80% of the cells are going to evacuate and die, all right, and go on, maybe carried by an animal to somewhere else where they can eat more food. All right, this is an incredibly dynamic process. All of these cells initially were clonal. They were treated the same. We actually started from one cell at the beginning, and yet now, 20 hours into this video, cells that were initially feeding are now 20% of them are dead. I mean, what happened here? How do you decide to go back and forth and figure out who's going to do this? How are they passing information between each other? Who's making that decision? Totally crazy. All right, so the networks that control these decisions in all systems, in all biological systems, are very complex. This is the one that's going to be driving this here in slime mold. And what we now do is go ahead and eavesdrop on the dynamics of key players in these networks and ask, what is the relationship between what's going on in the environment and what they're doing, and the relationship between the information they're using to encode this and molecules like second messengers and master regulators? How are they behaving to allow them to execute this program? All right, so in this particular system, they're using a small molecule called cyclic AMP to execute this. All right, we can now go ahead and eavesdrop on what the cells are thinking about what's going on as they execute that program. So in this panel, we're seeing fluorescently labeled cells, and here we're actually going to see those same cells, but we're now eavesdropping on their information coding inside of them. And so without doing anything, just by starving them, they start to execute this program where they send each other communication waves. Again, we're seeing inside the cells, but these are thought to be mirrored externally in waves of the molecule outside as well. All right, the waves are going to self-stabilize and begin to start to come from certain signaling centers. They're going to become faster and more intense. It's a little easier to tell over here, but around this time, you're going to be able to see the cells move pulsally in response to each wave. All right, so they're now executing behavior and not just using this to decide where to go, what to do next. They're now starting to go down here.
How fast is this sped up?
So this is hours, minutes, seconds, so we're now 9 hours in. So these waves, to give you an idea, their top frequency are about 7 minutes in period and about 3 minutes in width. All right, I just might think, what's going on here? Is this just allowing them to move into this system? Partly the answer is yes. The cell-cell communication is allowing them to relay a signal that tells them to move somewhere. But this is also actually used to coordinate gene expression and coordinate the decision to make that bizarre structure. All right, so since we're short on time, I want to sort of quickly summarize what's been done and give you more of the future-looking perspective. We're able to actually go ahead and use these technologies that allow you to eavesdrop on cell signaling, right, to understand what are the basic things a single cell does to coordinate cell responses. Right, these cells can modulate their internal behaviors in response to different levels of environmental signaling molecules. We can use that to build basic models, right, that recapitulate these phenomena. And to sort of get back to one of Eric's key points earlier, we actually have a lot of evidence that potentially noisy fluctuations in signaling, all right, heterogeneity, and that noise-induced heterogeneity are what actually allows this whole process to start and proceed. So I want to sort of give you an idea of where we can go with this. All right, I told you that this is a sort of natural system. They're going to build this structure. This is actually a real photo of slime mold in the wild. Something I think is super cool and a real thing that we could be taking advantage of a lot more in the lab is the ability to interrogate natural systems. So part of the challenge here, right, is this is the natural thing that's out of the dirt. All these videos I've shown you are with lab strains. We often work with some lab fancy organism that's been highly evolutionarily tuned in the laboratory to be able to easy to work with. And so how do we work with nature and really understand how does it induce heterogeneity and encode information differently to produce different phenotypes? So in this video, I just told you that the max signaling frequency is about 7 minutes. We can actually take a wavelet transform and see how these waves evolve over time, right? And it's actually highly characteristic in each genotype, how they behave, right? Something at the molecular, at the genetic level is encoding the rate of information transmission, right, and the information that's been able to encode. How do you look at this in nature? You can't just take stuff out of the dirt, throw some genes in it, and figure out how they're processing information. This is a real challenge. So something that actually people are working on is ways to do this. It used to be the state of the art was we could just sort of watch behavior and go ahead and see different strains signal and see their behavior on a sort of coarse-grained scale where we see the whole group send signaling waves. All right, but understanding if noise or different heterogeneity and dynamics is driving a whole system and allowing complex behaviors to emerge requires being able to eavesdrop on single-cell behaviors. All right, and so one of the places that I think is really really exciting and is going to change the field over the next few years is the ability to genetically manipulate natural isolates from the wild. So if you go ahead and take one of these coarse-grained videos, you can see they have a very different signaling phenotype than a lab strain. And due to the hard work of some really amazing students, we've just been able to make these genetically tractable. As in literally take stuff that just grows in the wild, eats bacteria, go ahead, we can stick genes in it and now have the possibility to watch the ways in which it aggregates and behaves differently. All right, so this is actually a wild strain from North Carolina. It's a little hard to see with these lights on, but actually it's a very different aggregation, completely different phenotype. All right, so we're now really excited and have the opportunity to go in and dig in and understand how do single cell properties change this in a way that allows them to behave differently. Another thing I think is really going to transform this field is the idea that we can do proof by control directly on signaling. If we can control information processing in a cell ourselves, right, we have the ability to understand what's actually causal. All right, so the idea here is that you could stimulate a single cell, induce a behavior like aggregation, ask what were the important information coding dynamics that it was using to discriminate between potential fates. So in this kind of system, we can actually steal stuff from tools from bacteria. This is light-activated cyclase. All that means is that this fancy molecule that's going to allow the cells to make cyclic AMP and send it to other cells. If we put two of these cells next to each other, we can see that they're actually able to communicate when I stimulate one cell and look for a signal in the other cell. In the interest of time, I want to only show you one video, but we can actually use this to create new behaviors. All right, through tuning their own information programs inside the cell, we can tap onto things that you don't see anywhere else. So if I go ahead and put about a dozen of those cells right here in the center, we can skip all that back and forth you saw in previous videos, get all the cells to aggregate into one center, and then actually tap into their own program that will allow them to crawl away as a group over a time scale of days. This is hours and minutes. All right, I'm getting the, you're short on time look, and I want to have a few minutes for questions at least. So what I want to do is I want to go ahead and skip most of the second part of my talk and just give a quick flash for some cool stuff we're doing in terms of understanding how groups of cells and tissues in your skin also signal and encode information about their environment, or potentially use this to decide how to heal wounds. So this is an ad for Come Talk to Me and go look at more fancy videos. Okay, that's another more fancy videos of this. We've got lots of them. It's very reproducible. But to sort of wrap up, come talk to me about, are these signaling dynamics causal? I think this is a super important question. Right? We can see a lot of information going back and forth in these systems now, and we have just enough tools to be dangerous. Right? And so I'm really interested in talking about, can we understand what's actually important for the behaviors we see executed next versus what's not? Two, for those of you who are interested in the actual biological alphabet, all the different parts of signaling pathways, we've got a lot of different tools that different pathways we can interrogate now to understand how information is being propagated from one signal to another. And then I touched briefly on two different systems. I'm super interested also in the motility switch in bacteria, which you've heard about before. So also come talk to me about how information is being used to potentially drive bacterial growth. So I want to sort of cap off on a couple of challenges I think are really important. What I hope I've been able to convey to you is that we now have the ability to quantify and control signaling across multiple multicellular organisms and systems. Right? You may have your pet one or the thing you're most familiar with, but this is actually a really broadly applicable set of tools we now have that allow us to interrogate and quantify. But I think we have a couple key challenges. I think the first sort of thing that we can sort of ask ourselves is what dynamics are actually important? All right, and is important just causal, or is there something else that makes something be important? Right, we're very good at seeing things and making a model that can recapitulate them and say, look, this could be what's going back and forth. Are we actually looking at the important information or not? And what are the tools that we maybe need to be developing or techniques we need to develop to discriminate between just inference and causality? And I hope we can learn something about the evolution and the optimization of cell-cell signaling and network architectures for information transmission and encoding. I think we now are also at a unique place where we're not just working with laboratory organisms, but now have access to a great depth of biological diversity. And we're just developing the tools across organisms to be able to interrogate these. And that's going to allow us to understand how, say, potentially a cell-cell signaling receptor could allow for the evolution of multicellularity or different amounts of information to be transmitted and thus different behaviors. We're also even understanding, are certain network architectures better or worse at transmitting information? What are sort of the important things about these biological architectures that we need to recapitulate in synthetic systems? Right, I think there's a lot of exciting questions there. All right, and I really want to advocate that there's a real future out there for really just watching what things do, quantifying those behaviors, modeling them, and then controlling them to really understand, are we looking at the most important dynamics in these systems? So with that, I want to thank particularly all the students who did this work, my collaborators, and everyone for the money to do this fun science.
We have time for a question while our next speaker comes on up. In the back, have the hand up quickly.
So that's extremely cool. So I was wondering, in the case where there's these different strains of Dictyostelium, if they, clearly have very different behavior, is that relevant to their, so in the lab, they do something different. I'm just wondering, is it possible to ask, do they build different structures that are evolved for their environment? Are their dynamics somehow matched? So how long it takes them to aggregate, how long it takes them to crawl or build a stalk? Is there any data on that you can sort of, as you were talking about, like find the kind of causal sort of basis for these differences in signaling and behavior?
Absolutely. So all of these things do exhibit very different behaviors on all of those sorts of timescales. So both the crawling, the structure size, how they partition themselves, how they compete with each other, those are all modulated in different genotypes. In terms of, I think, suitability toward environmental causality, we don't have a good idea of how to approach that question yet, in the sense that they're living in very complex environments. All we get is the sort of end result where we can go out and say, okay, look, there's genotypic diversity and these exist together. And so we do have nice collections to probe that question. I think that's possible. I think also there are some exciting projects that people are working on in terms of building indoor places to evolve microbes and see how. I think that's basically my answer is we don't have an answer yet, but I think it's on the horizon and a thing we can do.
Great, thank you very much.